we present for the first time a new approach for reproducible and scalable generation of iPS cell-derived cerebellar organoids under chemical-defined conditions using single-use bioreactors.
To obtain cells for seeding the bioreactor, add 1 milliliter of cell detachment medium to each well of a 6-well plate of human iPSCs. Incubate the plate at 37 degrees Celsius for 7 minutes until gentle shaking easily detaches the cells from the well. Using a P1000 micropipette, pipette the cell detachment medium up and down until the cells dissociate into single cells. Next, add 2 milliliters of complete cell culture medium to each well. This will inactivate enzymatic digestion. Then use the pipette to gently transfer the cells to a sterile conical tube. Centrifuge the tube at 210 times gravity for 3 minutes. Remove the supernatant and resuspend the cell pellet in culture medium. Count the cells using a hemocytometer and trypan blue dye. Seed the bioreactor vessel with the iPSCs at a density of 250,000 cells per milliliter. Insert the bioreactor vessel into the universal base unit in an incubator at 37 degrees Celsius, 95% humidity, and 5% carbon dioxide. To promote iPSC aggregation, set the agitation rate.